This is a production of Cornell University.
Really great. Thank you for joining. And today I'm going to be talking to you about my thesis titled Understanding the Role of Cellular Processes in Grafted Plants. So in nature, a process known as inoculation occurs when a process known as inoculation occurs in nature where different plants are forced together either by pressure or wounding, and it leads to the fusion of vascular or non-vascular tissue. And it's thought that this natural phenomenon may have inspired early humans to begin grafting. So grafting is an ancient process that's been utilized for at least 2,000 years, originally to propagate woody crops that were either difficult to grow from seed, did not grow true to seed like apples, or could not be rooted from cuttings. And today during my talk, I'm going to utilize consistent terminology. And so the way that I refer to the different parts of the graft tell us where that piece came from originally. So the scion, which is actually Old French for bud, is the apical portion, so the portion that's being grafted on top. And the stalk is the part that gives us the root system. And when I notate this, it's going to be scion, stalk. So this will be consistent throughout the whole talk. Grafting is used for a lot of economically important crops, including one of my favorite, coffee. So here I'm showing you coffee Arabica, which is the desirable scion grafted onto coffee coniferæ, which is a resistant rootstock. So grafting can be used to confer abiotic and biotic disease resistance, like we see here. And more recently, it's also used in the propagation of herbaceous crops. So cucurbits were the first that were used in this way. And in a lot of East Asia, almost all cucurbits are grafted to pumpkin rootstocks because it allows them to be resistant to certain, like fusarium wilt and other diseases present in the soil. Our lab uses tomato as our model system. This is because it has a great toolkit of genetic resources, including a sequenced and well-annotated genome, multiple expression atlases, and a well-established transformation protocol. In addition to this, it is also grafted. It is sold as a grafted crop. You can find it grafted onto many different things, including other tomato varieties that help to boost yield and some very fun kind of novelty grafts like you see here. So the Solanaceae family in general is often intergrafted. And during grafting, whether it's a woody plant or an herbaceous plant, it's going to follow a conserved timeline of physiological processes. This first starts with the initial adhesion. So this is when the scion and the stalk touch each other, and there's going to be a secretion of different sticky substances, pectins and polysaccharides that help the plant just adhere to each other. Next, a layer known as the isolation layer, shown here is this kind of little brown line, which is just cellular debris that gets kind of stuck there. So that's visible. And we also start seeing the proliferation of new callus cells. The callus cells are our undifferentiated Band-Aid that helps to hold those two pieces of tissue together. Eventually, we start to get enough cellular differentiation there that the scion and stalk are cellularly connected. Next, we have the non-vascular cell proliferation. So meaning the pith, the cortex, epidermis, that's going to start to differentiate. And we also get vascular tissue differentiation. Once we have enough vascular tissue restored, transport is able to reoccur and the graft is fully healed. And despite a really long history of using grafting, we only have a handful of genes that we've been able to show are directly related to grafting. And these genes fall into kind of these general categories of that initial adhesion step, the non-vascular proliferation, and the vascular proliferation. So I am specifically interested in graft compatibility, and anyone who has spent any time with me in the last 5 years has heard way too much about it. So graft compatibility is when two plants are grafted together and the xylem and the phloem is able to reconnect. This is in contrast to immediate incompatibility, which is when you graft two things together, there's immediate death. But what's very interesting is something called delayed incompatibility, where you can graft two things together and from the surface, it appears healed. It might even continue growing, but months or even years later, the grafts will fail, either due to failed transport or they'll break because they're just not adhered to each other well. Sometimes this is seen You can actually tell like this very unhappy woody plant. This is a overproliferated callus, likely due to pooling of photosynthates, or when the bark is peeled back, you'll see these demarcations where the xylem never healed. And there's a lot of different potential causes for this. And some of the hypotheses that are very abundant in the literature include the effect of phylogenetic distance, failed vascular reconnections, and also disrupted communication between the scion and stalk. And so today, I'm going to talk to you about 3 chapters of my dissertation that each address one of these potential hypotheses, starting with phylogeny. And this section is titled Understanding the Taxonomic Constraint in Solanaceae Graft Compatibility, Building a Graft Incompatible Model System. So it's been historically noted for thousands of years, actually, that more closely related plants are more likely to be compatible. And there's lots of exceptions to this rule, but in general, it's a good place to start. So this means that when you're looking at graft compatibility within a species, so different varieties that exist within the same species are very likely to be compatible, such as which is seen with apple. If you move up one layer to genus, there are certain genera that are very compatible, such as Prunus. There are many different graft compatibilities present within this genus. So those are interspecific graft compatibilities. But there's also some exceptions in there that aren't compatible. So it's not always true. And in our lab, we've been able to show that Solanaceae has a lot of compatibility intergenically between things like petunia, tobacco, and then also these intragenic grafts like eggplant and potato. And recently there was this very interesting paper that came out by Michitaka Notaguchi. This was in Science, where they showed that the presence of these beta-1,4 glucanases, or essentially very sticky substances present in Inbenth, allowed it to be compatible to a really wide variety of things, including closely related plants like tomato and very distantly related plants like Arabidopsis, which is seen here. And so I was interested if I could hone in on some economically relevant crops and see within this group of Solanoideae plants what the compatibility status was. So for this experiment, I looked at Capsicum annum, or pepper, Physalis pubescens, or ground cherry, Solanum lycopersicum, tomato, or Solanum melagina, eggplant. And these were a pretty appropriate group of plants to test because not only do they have very similar stature when we grafted them, meaning that they both have kind of long, thin hypocotyls, but the width or the diameter of the stem itself is very similar. So when these were grafted, they were all between 1.5, 1.75 millimeters. And this is important because it increases the chance that the cambium and the vascular tissue are actually going to line up with each other. So I grafted 16 different combinations, and I was actually able to get at least one plant to survive in every single graft. Obviously, there's some that performed better than others. For instance, ground cherry grafted onto eggplant was not super happy to be around 30 days after grafting. And when we look at each of these plants, so the x-axis here is showing you the scion, and the y-axis is showing you the stalk. Here's all the 16 graft combinations. We're actually able to see some signs of stress, including signs of drought stress, including rolling leaves or a little bit crunchy on the edges, brittle stems, so they're very fragile, or anthocyanin accumulation, which is a sign of stress in tomato. And really our first line of action whenever we're looking at graft compatibility is to perform a bin test. And the bin test is an excellent test because not only is it essentially free, it requires very little training, and it's relevant for grafting because grafting often happens in the field. So this is something that breeders and growers can actually test. So when you perform the bin test, you're going to hold the plant on either side of the graft junction and just give it a little gentle bend. So this is a self-grafted ground cherry, nice and stable. And the graft junction is this wide part. So it's unable to break. And actually the stem above is weaker than the graft junction. Similarly, this is eggplant on tomato. And when I give that a little bend, it breaks up above the graft junction and not at the junction itself. Whereas this ground cherry pepper plant is quite fragile. And when you give it the bend test, it's very clear that that graft junction was really not held together with anything very strong. So we can look at the rate of break that occurred in all of these different graft combinations. So the self-grafts are these first 4 columns, so they're very low. And then there's also 2 heterografts that are very low. So tomato eggplant and eggplant tomato, as well as all of the self-grafts, were very stable and unable to be broken, whereas all of the other heterografts were actually very easy to break. And when we do the bend test, what we're really doing is a proxy for how much stability there is in the junction. And we know that the kind of support beam of the plant is xylem. So when it's easy to break, that's a good indication that there is no xylem connecting those 2 pieces. So when we look at the anatomy inside of these graft junctions, we can actually see this. So the green boxes here show which of these graft combinations actually have a xylem connection, which I like to refer to as a xylem bridge. So xylem that's running across. If we look up close, this is a self-grafted tomato, so obviously very compatible. And the dashed line is showing you the graft. And this blue line is running right next to the xylem. So the xylem is very bright white here, very easy to pick out. And if we zoom in on this to make it really clear, you'll see that the xylem cells that span the graft junction are very cohesive, they're very organized, and you can actually follow a single file of cells all the way through. So that it's very coordinated. And so we see this with all of the self-grafts as well as tomato and eggplant. Whereas an incompatible graft, so it already looks a little different from here, is going to have discontinuous vascular tissue. So we can show that the xylem kind of approaches the graft junction, but they never meet each other. And if we zoom in on that, you can see that the point where maybe you would hope that the xylem connected is actually full of undifferentiated cells. So there's some sort of miscommunication that occurs that doesn't allow this plant to connect its vascular tissue. And we see this with all of the other heterografts. So we can kind of see some different things. For pepper eggplant, we see an overproliferation of cells. In the ground cherry tomato I showed you, we have undifferentiated cells blocking this, or you can have actually an air layer. So there was never contact and things just didn't heal. So we were able to show that tomato and eggplant, which are both members of Solanum, are intragenically graft compatible. And if you'll recall, potato is also compatible with tomato and eggplant, which is another member of Solanum. So Solanum seems to be very graft compatible. But all of the other genus that are in this experiment were not compatible with each other. So capsicum, physalis, and Solanum, I was unable to find any compatibility there. And this is actually very exciting because it allows us to generate models to study incompatibility and compatibility. So as a compatible model system, we have eggplant and tomato, and as an incompatible model system, we have pepper and tomato. And so my research really asked, can I utilize this incompatible system to better understand the genetic and anatomical processes that occur in compatible systems? So I'm going to use this system for the rest of my talk to address different questions. So if you recall that when I grafted all of these plants together, tomato and pepper kind of have a moderate level of survival. They don't perform as badly as some others, but they're, tomato pepper is not happy. And it's especially interesting that you can get kind of a diverse array of phenotypes when you graft these plants together. So here you've got pepper grafted on tomato and tomato grafted on pepper. And in the same exact batch of grafts with the same exact varieties, you can get plants that look like this. So there's a very interesting kind of diversity that we see there. And so I wanted to see how consistent this system was across varieties. So I tested for habanero, Dour de Landes, cayenne, and California Wonder. These are plants from 2 different species of pepper. And I chose habanero, cayenne, and California Wonder because they're representatives of economically important peppers. And I chose Dour de Landes because I have beef with this paper. So 40 years ago, this paper was published, and it actually lists Dour de Landes as a compatible graft partner to tomato. So I was really interested to see if this would hold up. So when I graft these plants together, I'm able to track their survival over time. So I'll talk you through the colors here. The yellow line is self-grafted tomato. So that's the same for all of them. The green line is the self-grafted pepper combination. So they always perform very nice, very nice. And the heterografts are these purple and blue lines. So there's a significant reduction in survival in the heterografts of these peppers. And then we come to my pepper of interest, Dur-de-Lanz. So this is a really interesting graft combination. So green line is self-grafted pepper. So this actually pepper grafts really poorly. It has a very low survival when it's grafted to itself. But interestingly, it survives pretty well when tomato is grafted on top. So I think that this is probably what got people thinking that it was a compatible graft. And so I needed to see if I could break those peppers. So I looked at the tomato pepper heterografts, gave them a little bin test, see what happened. Broken. So I think that they're just really good at sticking together, but it doesn't seem like there's any vascular connection there. And for the peppers that grafted very poorly, they had very poor survival. So this is self-grafted Dur-de-Lanz. Despite the fact that they survive at a very low rate, the junction itself is very stable. So this is likely just a pepper that behaves a little bit differently than the other ones, but I don't believe it to be compatible. And this is all of the other pepper graft combinations. I've generated this nice little montage to show you that I broke a lot of peppers for this graph. So I went through and despite very small stems or very fat stems, almost all of the heterografts were easily broken, whereas the self-grafts had a very stable graft junction. And if we plot survival against the percent that broke, so survival is the y-axis and the percent that broke is the x-axis, we of course have Dur-de-Lanz hanging out in the middle by itself. So kind of low survival, but also low break. All of the other self-grafts cluster up here, high survival, low break, and all of the heterografts have high break, low survival. So this seems to be a pretty stable trend with the exception of Dur-de-Lanz. And I wanted to see if, we've now said, yes, they can be broken or no, they can't. I wanted to mathematically quantify this. So to do this, we collaborated with Dr. Erin Sparks and her PhD, who is now Dr. Lindsay Irwin at the University of Delaware, to do 3-point bin tests. The way that this works is that a piece of tissue is placed inside of an instron on 2 fulcrum points, and the 3rd point is the force probe. So those are the 3 points. And here is my tissue in there. And so as the instron pushes down, it records the force required to displace the tissue. So that's what this chart is showing you. This is the force displacement curve. And we were very interested in this elastic measure, which is the amount of force required to displace the stem prior to break. There's a few reasons we were interested in this. It's a good measure of kind of the elasticity or the bendability of the graft junction. And it also can be mathematically converted into the bending modulus. So the bending modulus is a unit of measurement that allows us to record force or tension regardless of the structural properties of what we're comparing. So, tomato and pepper are structurally dissimilar. They have different cellular composition, and they also have different size. So by converting this to the bending modulus, we're able to directly compare all of these samples. And what we see is that when we exclude size from the equation, there is a significant difference between if a plant is ungrafted, self-grafted, or heterografted. And if we then look at each individual sample, we see that the heterografts are significantly different than self-grafted tomato. They're less, but they're kind of similar to self-grafted pepper. So we were able to show that there actually is a reduction in the amount of force required to bend the heterografts. And in some of these comparisons, it's very significant. And again, we had to look at the xylem. So here's self-grafted tomato, looks beautiful. This is self-grafted cayenne, looks very similar to self-grafted tomato. And in the heterografts, the first picture shows you a scion tomato. The second picture shows you tomato as the stalk. So you'll see that there's some disruption in the vascular tissue. You might see these over-proliferated callus bulges, and the plants themselves look pretty puny. So we see this with all of our tomato pepper graft combinations, very likely to see callus over-proliferation and even adventitious roots, which is a symptom of graft incompatibility. So the scion is just like looking for water, so they throw out roots. Like I mentioned, the plants are very different in size. So this is self-grafted tomato 30 days after grafting, And oh, just in case you can't see it, there's tomato pepper 30 days after grafting. So really quite reduced. I quantified this as well. So I measured the area of the scion and the area of the stock. I was able to show that this was super significant. Depending on if it was a compatible or incompatible graft, the amount of growth that occurred was very different. This is likely due to just reduced transport of nutrients and water. I also measured the lateral growth that occurs, so how much the stem was able to grow in 30 days. This was also significantly different depending on if it was compatible or incompatible. So I've been able to show that across all 4 of these graft combinations, we have consistent vascular disconnection, junction instability, and reduced growth. So we're able to use this model to ask other questions. So next we'll talk about vasculature, with this title being gene regulatory networks for compatible versus incompatible grafts, identify a role for WOX4 during graft formation. So we don't know that much about what's going on in the stem of a grafted plant when it's redifferentiating the vascular tissue. But what we do know a lot about is what happens in the root of Arabidopsis. So this is a good place to start. And what you see here is that there's a lot of signals that are expressed in one cell and then act non-cell autonomously in a different place. So that means that there's movement and coordination that has to occur. And we don't know if these signals are moving apoplastically or symplastically. Some of them we know, some of them we don't know. So there's a lot of open questions on how the vascular tissue is formed, especially in the stem. And I've showed you that self-grafted plants heal the vascular tissue, while these heterografted plants don't. These heterografted plants have a very uncoordinated kind of discombobulated xylem formation that implies some sort of vascular signaling breakdown. So we wanted to see when this happens. To do this, we looked at grafted plants 3, 4, 5, and 6 days after grafting. Here I'm showing you self-grafted tomato, where the callus is signified by little yellow arrows, newly formed protoxylem is marked with green arrows, and the xylem bridge, or the continuous xylem strands, are red. So in tomato, we have lots of callus and protoxylem by 5 days after grafting, and by 6 days after grafting, we have continuous xylem. In pepper, we see something similar, albeit slightly delayed, where we see a callus and protoxylem proliferation at 4, 5, and 6 days after grafting, and not shown here is xylem connection 7 and 8 days. Whereas the heterografts have tons of callus and protoxylem proliferation, but a failure to achieve xylem bridges. We see that in both tomato pepper and pepper tomato. So we were able to show that the tomato pepper and pepper tomato vascular dysregulation occurs in the first week. We wanted to see if we could understand the genetic regulatory process that underlie what we see. So we looked at self-grafted tomato, self-grafted pepper, and the heterografts at 3 time points that represent important physiological steps in graft formation. So adhesion at 1 day, callus formation at 3, and vascular differentiation at 5. And before I talk about this, I want to give a huge shout out to our collaborators at North Carolina State University, Dr. Lisa Vandenbroek and Dr. Ross Cezanne, who not only built and published the software that's used to generate these networks, but they were also very important collaborators, and this work wouldn't have been possible without them. So the data I'm showing you here today is the compatible grafts. And if you're very interested in the incompatible grafts, I'll talk just a little bit about that in a minute. So in the compatible grafts, we're able to use our transcriptomic data to generate Bayesian networks. So these are dynamic Bayesian networks, which specifically is utilized when you have temporal data. So if we kind of zoom in on what these center parts, so the big yellow dots are the hub genes, which are genes that are predicted to regulate many downstream genes. And the color of the line coming off of them tells you what day they're predicted to act. So green lines are 1 day, blue lines are 3, and red days are 5. So there's actually a ton of information just in these networks. And I want to talk to you all about it for an hour, but I don't have time. So if you would like to check out these networks and shoot me an email, I'm happy to talk about these. And the incompatible networks are also in the paper, so I encourage you to go check those out as well. So I'm going to show you a different way to look at these self-grafted hub genes. So if we start in this middle column right here, The gene IDs that are listed here are the hub genes. If you move one column to the left, you have the transcription factor family that that gene belongs to. And one column to the left, you have the graft combination that that hub came from. If you return to the middle, one column to the right, you have the time point that is predicted to act. And one more column, you have different go terms that these genes fall into and their predicted role in grafting. So what's really interesting here is that we expected that tomato and pepper would have many orthologs of the same genes functioning in this process. But what we actually found was that tomato and pepper use, in a lot of cases, completely distinct transcription factor families during grafting. So you'll see all of these guys over here are only attached to tomato, and these guys down here are largely only attached to pepper. So we were able to show that different species can utilize completely distinct genetic pathways to accomplish the same goal, which is grafting. And at first this was surprising, but after I thought about it, it made a lot of sense because grafting is not an evolutionarily conserved process. It is humans taking advantage of plants' robust wound response and kind of hijacking that for our intention. So it makes sense that plants would have very diverse ways to handle wounding, and that it's not necessarily conserved in specific genes. So I talked to you first about how tomato and pepper heterografts have this vascular disconnection phenotype, and then I showed you these networks. And in these hub genes, if you have a chance to look at them later, you'll see that most of these are actually not related to vascular differentiation. They're related to non-vascular processes. So why does this happen? It kind of highlights a shortcoming of Bayesian networks, which require the genes in your data set to be highly abundant in order to be represented as hubs. And we think that the reason we don't see vascular differentiation genes highly abundant is because the cells that are being differentiated make up less than 1% of the graft junction. So here I'm showing you 5 days after grafting, and these newly formed protoxylem are highlighted in that light pink color. And it's a really small percentage of the tissue that was actually ground and RNA was extracted from. So in order to capture this specific subset of genes, we returned back to Arabidopsis, and we selected this subset of genes that are involved in xylem cambial maintenance. We identified orthologs in tomato and pepper utilizing OrthoFinder, and we wanted to see if they were disrupted between the self-grafts, which are down here, and the heterografts, which are above it. And what you can't really see just by looking is that every single one of these genes is significantly disrupted at least one time point. So we put these genes into a regression network, which is a little bit distinct from the dynamic Bayesian network because we're utilizing one time point and utilizing second time points to infer the direction of it. And we were able to identify WOX4 as a highly connected gene within this network. And we are super grateful that Dr. Zach Littman at Cold Spring Harbor was willing to share a WOX4 mutant with us. And if you remember, there's WOX4. In Arabidopsis, it also has a redundant ortholog, WOX14. In tomato, WOX14 is not present. So we expected that because WOX4 is upstream of cambial maintenance, that there would be some significant difference when we grafted it. First, we wanted to check out the mutants because they hadn't really been analyzed yet. And we found that the WOX4 plant was slightly smaller. It had a reduced leaf size and reduced root size, but that the vascular arrangement inside of it, so the xylem are these very large kind of blue stained cells down here. They look pretty similar. There's not any like severe mutations or really strong phenotypes. So we were confident that there wasn't anything largely wrong with our plants going into grafting. The maybe most disappointing graph of my PhD was when I grafted these and they all performed almost exactly the same. But luckily, I knew that I could perform a bin test on them. And I showed that the self-grafted WOX4 plants were very fragile and easy to break, whereas the wild type plants were very strong. So despite there being no difference in survival, there was a huge difference in the integrity of the junction itself. And then when we looked at the xylem in these, we found that self-grafted wild type, as well as the heterografts, have beautiful xylem bridges, nicely connected vasculature, whereas the self-grafted mutant has a very kind of chaotic and disorganized graft junction that kind of phenocopies what we see in tomato and pepper. So we were able to show that WOX4 is required in grafting in tomato, but it actually has no directionality. So that means it can be present in both sides of the graft, or it can be present in just one, and that's enough. And there's lots of open questions on WOX4, and I think you'll see more of it from the Frank lab. So we're really excited to be able to drop WOX4 onto this figure, especially because it's the first gene that was identified in an agronomically grafted crop. So it's very relevant. And have no fear, these networks are well underway in the Frank lab to identify new tools. So Dr. Greg Martin had a PTI5 mutant that he was willing to share with us. And if you're super interested in that, Alice in our lab is going to be presenting it as her honors thesis. So stay tuned for that email, attend to learn more. Audrey Woodmeyer has been working tirelessly to help generate these lateral boundary domain mutants. And Alice and Audrey have worked together to generate this ERF4 mutant. Additionally, we're working on the genetic tools as well. So Zach Littman, in addition to WOX4, shared the tomato ortholog of cleave 41, 42, and 44. I've generated an expression fusion, or an expression gene, and a fusion line in tomato. So those will be exciting for the future. And Brandon in our lab has been working to generate pixie mutants, which we hope will have a very severe phenotype. So more to come from that. And lastly, I'm going to talk to you about communication. So this is an exploration of tomato pepper's graft junction isolation layer. So the graft junction has kept me up at night for 5 years. And that is not because I dislike it, but because it is a very interesting anatomical feature with this dynamic intercellular communication and tissue differentiation. And if you recall that in the process of grafting, this isolation layer forms. So this is debris that's just left over from the process of grafting. And over time, this debris is going to be cleared from the graft junction, getting less and less and less until it's finally gone. And it's important that this happens because we believe that when the scion and the stalk make that first contact, they have cellular contact, that's when mobile signals would be able to be transmitted from scion to stalk and stalk to scion. And we know that in grafted plants, secondary plasmodesmata can form at the graft junction. So it's likely that there is some sort of mobile signals being transmitted at this point. And if this isolation layer remains there, it could be a problem. So because these really gross dead tissue layers are stuck in the graft junction, they could be a physical barrier to the communication that's required. And because this persistent death there is a symptom of incompatibility, I thought it could be a potential cause. So Margaret mentioned I came from a rat lab. I actually came from a rat immunology lab. So you're gonna have a crash course here. This is really kind of the inspiration for why I've thought about this project. And it's because in animal systems, when a foreign tissue is introduced, it's gonna give off antigens. These antigens are a signal of non-self that T cells recognize, transmit to B cells, which create memory. In addition, these plasma cells are going to be putting out antibodies that then tell the cell itself, what is this thing? Attack, and that's rejection. So I was curious if something similar to antigens might exist in incompatible grafts. And this really got me thinking, what does it mean for a cell to die anyway? So I've got these 3 little cartoons of cells. So we've got a healthy cell with a nice, robust cell wall, and we have cells that are undergoing programmed cell death and necrosis. So they both have a compromised cell wall, but they have different appearance and different causes. So the difference in appearance is that programmed cell death causes a shrinkage of the plasma membrane, while necrosis can cause either no change or an explosion of the plasma membrane. So it can swell up even within the cell wall. And the causes of these are different. So it's hidden, but it says moderate stress. So moderate stress can lead to a signaling cascade, some sort of perception of that stress that then leads to a genetically controlled DNA degradation process. Whereas necrosis is caused by an acute stress or something so significant or abrupt that the plant cell has no ability to respond to this, and it leads to uncontrolled cell death. And these 2 types of death can be used as proxies to ask other questions, which is there intercellular communication that between the scion and the stalk that leads to that cell death? Or is this stress-induced death, which I have related to during my PhD. But so I wanted to ask, is there a compatibility signal that cells use to perceive self or use to perceive non-self? So to do this, I first needed to figure out what was dead in my graft junctions. So I used a viability stain called trypan blue that can enter into cells with compromised cell walls. So regardless of if it's undergoing programmed cell death or necrosis, the cell is able to enter it. And to do this, my wonderful undergrad Alice and I looked at ungrafted, self-grafted, and heterografted plants at 7, 14, and 21 days after grafting. Here they all are. And trypan blue is really interesting because originally it was used in kind of like single-celled organisms or cell cultures. And in that instance, it's very black and white. It either stains something blue or purple, or it doesn't. Viable, not viable. But in plants, which are these multicellular, deep tissued organs, you're going to have a spectrum of stain. You're going to have regions that are this kind of almost light blue, white color, or you have very little cell death, or you have regions that will stain back almost black. And this is because there's many, many layers of cells that are non-viable, so it looks black. And we have a similar spectrum in my work. So I modified this protocol slightly, so mine comes back a little bit more purple than blue, but that's okay. So areas that are containing very low levels of cell death stain this kind of light pink color, and areas that contain what I'm calling deep tissue cell death are this almost black purple color. So at 7 days after grafting, ungrafted tomato and pepper obviously have very low levels of cell death, whereas all of the graft combinations have these very deep cell death pockets right at the graft junction. And I'm pointing those with little yellow arrows. By 14 days after grafting, the cell death has reduced in the self-grafts, but it's kind of hanging out in those heterografts. And 21 days after grafting, it is pretty much gone from self-grafts, but still very present in those heterografts. And if we look at this as a percentage of the area of the graft junction itself, we can track it over time. So as we move through time, the percentage of cell death in the self-grafted plants reduces. And despite the fact that these pie charts look identical, they are not, I swear. The amount of cell death present in the heterografts is extremely consistent. So this is a persistence of cell death in the graft junction of the heterografts over time. So now I know that there is cell death in the junction, but I want to see what's causing it. Is it intercellular communication, or is it stress? So to do this, I utilized an assay called a tunnel assay, and it's called that because its real name is the terminal deoxynucleotidyltransferase DUDP Nikin labeling assay. So tunnel's a better name. And the way that this works is that the TDT enzyme can recognize double-stranded breaks present in DNA. These double-stranded breaks are really a hallmark of endonuclease cleavage, which is the way that programmed cell death occurs. These endonucleases cut up the DNA. And the TDT identifies the free hydroxy group and adds this super stable DUTP with a fluorescent probe stuck to it. So then when we image these under the confocal, only cells undergoing programmed cell death are going to be tagged with this fluorescent fluorophore, despite the fact that the cells undergoing necrosis are also dying. So we're able to differentiate between any cell and programmed cell death. An undergraduate who graduated from our lab last year, Ali Hermansen, really optimized this protocol. So she did a great job. And what I'm showing you on the bottom is just a little slice of a pepper stem that I've treated with DNase-1 to kind of mimic what a high level of those double-stranded breaks would look like. And in my work, I also counterstained all of this with propydium iodine so that we could orient ourselves inside of the tissue. But for the rest of my slides, I'm not going to show the propydium iodine just for time. You'll have to believe me when I say that we are, in fact, in the graft junction. So here are ungrafted stems at 7, 14, and 21 days after grafting. So these really bright strands that you see are autofluorescence coming off of the xylem. So they can be kind of distracting when you're trying to look at these. So it's like looking at stars in the night sky. Look at the black part of the sky and the stars will become more visible. So if you look over on these sides, you'll start to see these little puncta that are scattered randomly within the cell. And that's just the normal low level of programmed cell death that's occurring, probably not actually in the natural state, but of a plant that's just been chopped in half. So that's what we see. At the self-grafted plants, we, at 7 days, see kind of a low, normal-ish level amount of programmed cell death at the graft junction. But by 14 and 21 days after grafting, programmed cell death is starting to be elevated in the tissue surrounding the graft. So not just in the periphery, but also near the vascular tissue. And I'm going to show you a zoom up in a minute. And in heterografted plants, we see the same thing. We see an elevated level of programmed cell death in the graft junction as early as 7 days. So here's a zoomed up version of what you're seeing in those little pictures. These little puncta are cells undergoing programmed cell death. And what we found is that this elevated rate of programmed cell death is present in all grafted plants. It's not specific to if the plant is incompatible. So it's likely that programmed cell death plays some role in the process. And I've kind of hypothesized that it has to do with the transition of all of this callus differentiating into, whatever type of cell type it needs to be. And there's a little bit of cell culture work to back that up. That looks good. So deep tissue cell death does not overlap with the programmed cell death present in the graft junction. So the programmed cell death is present not only in the callus on the edges, but also in the tissue near the vasculature. This is not what we see in the deep tissue cell death, where these areas of death are very, very consistent on the outside. So the death that we see in tomato pepper grafts is not due to programmed cell death, despite the fact that programmed cell death is occurring in the graft. It is necrosis. So that means that some acute stress is acting upon those areas of deep tissue cell death, causing uncontrolled cell death and leading to stress-induced death. So the kind of outlying question is, what is the stress? So there's some different hypotheses here, and I have my favorites, but it could be metabolic toxicity. It could be a cytotoxic accumulation of ROS. It could even be osmotic stress. Any of these are things that can cause stress-induced necrosis. And so, at the end of all of this, I can say that I've generated a lot of knowledge about graft incompatibility in each of these hypotheses. So under phylogeny, I was able to show that there are many intergenic incompatible grafts in the Solenoideae, and most importantly, that survival does not equal compatibility. Also, I showed that egg mato and pet mato are ideal systems to study graft compatibility related questions. Under vasculature, I was able to show that tomato and pepper utilize distinct genetic pathways to graft, that incompatible grafts have perturbed gene expression, and that WOX4 is required for grafting in tomato. And then under communication, I was able to show that incompatible grafts have persistent cell death, that programmed cell death is upregulated in all types of grafting, and that the tomato pepper junction cell death is due to acute stress-induced necrosis. So next, I need to thank some people. Obviously, I need to thank the Frank Lab, which when I joined was just 3 people. It was really fun to watch us grow to a nice big group now. And even my favorite member of the lab, Guinevere, it was fun to watch her also grow up. And she doesn't even look like this anymore. She's so big. Also, a big shout out to Margaret. So if you don't know, Margaret runs, her whole organism is running off of gimme coffee and candy at any given time. That's all that's in her system. So I hope that after I've graduated, you can improve your pre-workout snacks a little bit. I think things can get better. I also want to say thank you to all of the research mentors I've had before this point. So down in the bottom left is Dr. Neil Snow. I think he's in the call somewhere, and Dr. Virginia Ryder, who were my undergraduate mentors, Blake Myers and his postdoc TC, and also the Van Eck Lab. Here we are as GFP, the nerdiest Halloween costume. I also need to thank all of my friends who are really important during grad school. It's good to have a support system. These people they're all over the world. Not everyone is here, but I'm really grateful to have had their support. Also my family, both those that I was born with and those that I married into. You've all been just like a backbone, a pillar, always asking me how I'm doing, Jayo, the whole time I've been studying, so it's been great. And then to Leo, who I forced to eat a lot of cheese, dress up as stupid Halloween costumes, and live through 400 inches of snow. So thank you for always following me to crazy places and doing strange things with me. And lastly, you guys knew it was coming. A huge thank you to my fur babies, Tater Tot and Sebastian, especially during quarantine when I was trapped in my house. It was so important to have them there. And lastly, there's a few other people I want to just specifically thank because they were very integral to my work. So Alice Gavorgian, right here, the best undergrad, Ali Hermanson, who graduated last year, my lab in-laws in the vitamin Nuke lab, who have let me steal probably more coffee than I want to think about. My committee members, Elena, Eric, and Joyce, the collaborators who were directly involved in the data I've showed you today, the staff at Cornell, including greenhouse and growth chamber staff, the plant transformation staff, and the imaging facility. All of you are really the backbone of the research that's done here at Cornell, and I hope you know that and get a lot of thank you. And my USDA predoctoral fellowship, which was a great thing to receive and to be able to focus on my research and to travel and do things with that. So with that, I'm happy to take any questions. Any questions? Yeah, sure.
Thanks so much for the talk and congrats. I was just wondering, you generated some gene regulatory networks. Did you happen to see any other, I'm thinking of like non-coding RNAs that popped out that helped regulate that tissue-specific or cell-specific grafting success?
So the question was if in my networks, I identified any non-coding RNAs and if those were involved in anything. So one of the assumptions you have to make when you're doing gene regulatory networks, at least in Bayesian inference, is that you have to be able to make the assumption that each gene is acting on the next gene. And so a really safe bet is to include transcription factors only in that original setup of your genes. And the reason why this is important is because Bayesian networks become less accurate the more genes that are present. So you have to make some cuts to keep the gene size small. So unfortunately, we weren't able to do that in our analyses, but I mean, the data is there. We have the RNA-seq data. So if you'd like to collaborate, I think hit Margaret up. Is there any?
There was a question.
Let me see. Nope, just go, Hannah. What are the biggest cases of graft incompatibility within the industry? Yeah, this is a great question because grafting is economically relevant. I think that some of the most probably economically important cases of graft incompatibility are happening within stone fruit. So there's lots of intergrafting that occurs between pears and apples and between plums and peaches, and not all of those are compatible. And in really confusing instances, sometimes they're only compatible in one direction, like plum has to be the scion and peach has to be the stalk for it to work and things like that. So that's probably one of the most broadly important, but I think it's more common that graft incompatibility occurs in like ornamental trees. And actually, if you walk around on campus at Cornell, you can find some that are incompatible. And oftentimes people will just sell those because they look good ornamentally for a while, and then eventually they'll break. So I think it's more common in ornamental, actually. Yes, definitely ask questions.
I have a question.
Sure.
It's just kind of like a hypothetical question. You don't have to have an answer. But if you were able to like tamp down whatever acute stress you think is causing the cell death, do you think that would then make them compatible, like incompatible grafts compatible? Or do you think that all of that cell death is like a side effect of the acute stress and like the stress is causing another thing that causes the graft?
Yes. So the question was, if we could reduce the stress that causes cell death, could we overcome graft incompatibility? I think in tomato pepper, the answer is no, because I think that there's two things happening. You have disrupted vascular kind of organization that's occurring. It's possible that the signal we're looking for is being blocked by the cell death, but it's also possible that there's something else going on there, like it could be a dosage thing, or it could be divergence of receptors or ligand, but we won't know that until we're able to figure out exactly what the stress is. and see if we can reduce that stress and then see what happens. So there's a few more experiments we need to do before we'll know for sure. Let's see. So someone asked, do you know if there's any effect of the geometry of the contact to flat surfaces versus a groove? Yes, this is a really good question. So something that happens when you graft is that there's an initial dry down period. So the tissue pulls back slightly, and each tissue pulls back differently. So tomato stays pretty cylindrical, whereas pepper has a really intense dry back. So ways that we overcome this is that instead of performing a flat graft, we perform a super steep slant graft, so that despite the pullback, the central portion of the stem is still in direct contact. So we've been able to overcome that, but there is a whole area of very horticultural graft work that has to do with optimizing the specific shape of the graft to improve graft success. So I won't say that I'm an expert in that, but I think we've overcome that. Commercial tomato rootstocks have complex genetics. How do they end up having such wide graft compatibility? Yeah, this is a really good question. So if you have very diverged plants, how are they able to graft? And so this is one of those exceptions. So, thousands of years ago, ancient Greek philosophers were writing about how plants that are closely related are going to be able to graft. And they were like very black and white in that, and plants that are distantly related won't. But we know that that's not the case. So whatever it is that the plants are recognizing in each other, or maybe it's a lack of recognition in those very distant grafts, whatever that is, it's not necessarily tied to how closely related they are. It could be tied to how much evolutionary time has passed between two things. Like maybe something has diverged in one species that has not diverged in another. And because we don't know exactly what we're looking for yet, we're unable to really answer that question well, but I've thought about it a lot. Yeah.
So with the incompatible grafts, it seems very interesting because they can grow for quite a bit of time until they break. So is breaking the only symptom? Because I'm thinking about like the trees around campus you're talking about. Like, would you see differences in like transpiration, or is manually going in and trying to break it the only?
Yes. So the question was, in incompatible grafts, is there anything else that happens besides breakage? So there is some work where they've done nutrient profiling in the scion, as well as checked turgor pressure using pressure bombs and things like that, which I dabbled in, but I didn't show you the data. And in the scion, when you have poor vascular connection, you have significantly reduced nutrient transport. And this hasn't been tested in everything, but that can lead to things like blossom end rot and issues with yield because you have low transport. And I would say that probably in the next 50 years, all of these incompatible trees we see on campus will break during a storm. So they will have some sort of wind or anything that stresses it, and it will break. And this is a really big problem for growers because the grafting is expensive when it comes to farming, because you have to train someone to go out and graft, either bench grafting or field grafting. And it's a really high economic and time input, with the expectation that that plant will grow fruit for maybe 50 years. And if it breaks in 4, as a tomato, something growing for 4 years seems like, oh, that's great. But as a tree, if it only grows for 4 years, that was a failure. So we're using an herbaceous model, but you have to think about that in the woody grafted world, it's a very different process. Let me read carefully. So they're asking, there's a lot of similarity between the networks and abscission genetics. In grasses, could be simpler to try to take a more mechanistic approach in terms of thinking the life history of cells and graft sites, the genes that regulate these, and if this could cluster into a suite of maybe 2 or 3 more or less optimal strategies. Yeah, I'll have to think about that. I think I'll have to think about that one. And I know, John, that we follow each other on Twitter, so I'll probably reach out to you after I've had a chance to ponder that and let you know what I think, because I haven't thought about the overlap between abscission at all. Yeah.
Do you know where WOX4 is expressed, like the cell chart maybe?
Yeah, so the question is, do we know where WOX4 is expressed? Kind of. So we know where WOX4 is expressed in tomato, but not where it's necessarily expressed in the graft junction. So there is always a kind of assumption we make that things are going to be present in vascular differentiation of the stem as they are in the root, or WOX4 is identified in tomato by the Scanlon lab. So Thanks for that. So we don't know that yet. But I think in the works is that we'd like to have a reporter line, or we've even had reviewers ask for in situs, which we politely declined doing for our publication. But we'd like to see exactly where they're being expressed, and also if they're mobile, because I have a lot of interest in that, and seeing what the dynamics are of WOX4 in the graft junction itself.
Oh, I just wanted to add to that. So in the class that everybody left to go, we were reading a paper today about tomatoes, and they were localizing WOX4 in tomatoes, and they found it in like what they call the phloem parenchyma, or the fibers. But I actually thought they looked like more pericycle, and I understood actually the WOX4 to be more related to cambium maintenance, actually, instead of actual like a differentiation of the cells.
Yeah.
So that's why I was wondering.
So the question was, there was a recent paper where they showed that WOX4 was possibly in the pericycle. Is that what you were?
I think it's pericycle.
Pericycle.
Phloem parenchyma.
Okay, so it's in a non-vascular tissue associated with the vascular system. It's interesting you asked that question, because I actually just yesterday was really thinking about the pericycle in tomato, as one does, and some of the papers I was coming across, people were making the argument that the pericycle should be considered meristematic tissue because of its ability to produce adventitious roots. So it's possible that in tomato it functions slightly different than what we're expecting. But we also, knowing that it's in the pericycle is very exciting, because one of the other graft-related genes, ALF4, aberrant lateral formation 4, directly related to pericycle maintenance after grafting. So, I'm not sure exactly what it's doing in the graft junction, but I'm happy to know it's at least associated with the vasculature and possibly associated with the tissue that some consider to be meristematic. So that's all good and tracks, and I'm excited to see where we actually can find it. Okay. Yeah, hopefully if other people had questions online that I didn't get to, please shoot me an email or a Twitter message. I'm happy to talk.
Awesome. Thank you for that wonderful presentation. Your committee's going to have a lot more questions, so we'll take a pause. And later today, we'll be on the second floor of plant science.
And a graduate in Cellular.
Thank you so much. It was great.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.